workshop. So I think we can get started. My name is Rebecca Batorsky. I work as a bioinformatics scientist as part of research technology, which is part of Tufts Technology Services. So I'm so glad that there's so much interest in this software. Over 40 people registered for the workshop, and that's great. As people know, we've started a license last month, and the license will go for a year. We'll be looking at the usage and also feedback over that time, and hopefully there'll be interest and funds to have the license renewed in the future. Today we have with us Helen Shearman, who's a PhD application scientist with Geneious, and she's going to walk us through some of the functionality. Some people requested specific topics to be covered, which was excellent. I do want to note that users are welcome to reach out to myself or to the Geneious support team if they have follow-up questions about the license or about usage of the tool. Geneious can be reached at support@geneious.com. I'll put that in the chat. And I have some information also on my website, which is sites.tufts.edu/biotools, which I'll also put that in the chat. I'm going to be muting everyone who's not Helen, just to make sure that we can all hear. If you have any problems unmuting yourself to ask a question, feel free to use the chat function either to me or to everyone, and we'll make sure that we get your question answered. So I'll be keeping an eye on that. I believe that's it. Please take it away. Thank you, Helen.
Okay, great. Thank you. So hi, everyone. Welcome. My name's Helen. So today I'm going to give sort of a general overview of Geneious, tell you a little bit about how it works, just where things are, and then we're going to talk about some of the various functionality, some of the various tools available. As we were saying earlier, you don't need to follow along with Geneious on your computer. You are welcome to if you've got it open, but you don't need to. The session is also being recorded, so if you want to go back later on and work out what buttons we were using, you'll be able to review the recording. The other thing I'll mention is I do like to keep these sort of informal. I know we've got a large-ish group, but if you've got any questions as I go, let me know. Either speak up. I'll try to stop periodically for questions. You can also speak up as I'm going, or let me know through the chat function, and we'll keep an eye on that. But with that, I've got just a couple of slides to start out with, just to tell you sort of what Geneious is, particularly for those of you who may be a lot newer to it, and some of the basic background information. And then the rest of this is going to be a live demo. So with that, like I said, just a couple of slides to start out with. And the first thing is sort of what Geneious does, because there's a lot of different tools, a lot of different analyses you can do, all from within this one piece of software. So from Geneious, you can search sequences. You can look inside a sequence, find areas of interest, like finding repeats or finding motifs. You can search against external databases, like NCBI or UniProt. You can also run BLAST searches, either against NCBI or against local custom BLAST databases. So if you have a particular genome you're working with, or a collection of genes, or maybe it's proprietary sequences, you can create a local BLAST database in Geneious and search directly against that. There's lots of tools for annotation, so you can create annotations, you can import them. There's ways to automatically transfer annotations between a sequence as well. So you can create like a custom annotation library in Geneious and use that to annotate new sequences. There's lots of different tools for cloning. So Geneious will do primer design. It will create primers. If you have a library of existing primers, you can import them into Geneious, see if they match against any sequences. You can do classical restriction cloning. You can do Golden Gate cloning, which is really just restriction cloning with type 2S enzymes. You can do Gibson assembly. And just be aware, when we talk about Gibson assembly in Geneious, that particular tool also does things like infusion cloning and CPEC cloning, some of those homology-based cloning methods. You can also do gateway cloning, topo cloning. You can also find and evaluate CRISPR sites. So lots of different cloning methods. You can also work with Sanger sequences or NGS data in Geneious, so bring in your ABI files or your FASTQ files. do pre-processing, so things like trimming off bases based on quality scores. For NGS data, there's some additional tools like error correction, normalization, deduplication, kind of those data QC steps. And then you can do de novo assembly, or you can map your reads to a reference sequence. And then you can do downstream analyses like finding where any variations or SNPs are. You can also do basic RNA-seq expression analysis. You can also do alignment, so pairwise or multiple sequence alignment, either with DNA or protein sequences, as well as building phylogenetic trees. And that's, in fact, actually the space that Geneious started out in something like 15, 20 years ago at this point, started out in that alignment and tree building space. And then finally, workflows. Workflows are my personal favorite tool within Geneious. They're a really great way to automate your analyses. So if you find you're doing the same thing again and again and again, even if it's something simple like building alignments and building a tree, then you can create a workflow in Geneious to automate that for you. Or if you want everybody in your lab to analyze your Sanger sequences the same way, or maybe you've got interns that you want, or students that you want to analyze the Sanger data all in the same way, you can create a workflow that does that. So they're a very powerful tool within Geneious. What I would say is get familiar with sort of the tools in Geneious first, and then once you're comfortable with it and you find, you know, there's certain things you're doing again and again, definitely take a look at workflows at that point. So that's kind of the high level overview of what Geneious can do. Like I said, Rebecca sent through some topics in advance, so we're going to talk about a few different things here, things like annotations, working with primers. We'll talk a little bit about alignments and trees, as well as some NGS analyses with some SNP calling. Given the time and how many topics, I'm not going to be able to go in depth into everything, but I will show you where you can find more resources as well, so things like tutorials. And like I said, you can follow up via send myself an email or send the support team an email. Rebecca shared that email address in the chat function. I'll also show you where to find it on our website. But also, like I said, if you've got any questions as I go, if there are any other topics here you want to talk about, just let me know and I'll try and cover those as well. Like I said, that's kind of the high level overview. And then there's a couple of other sort of basic background information I just want to cover. So Geneious is continually evolving. We release a new version about once a year. So the latest version came out in November of last year. So that's Geneious Prime 2020. And there was an update to that back in, I think it was March. So Geneious Prime 2020.1 is the latest version. Your license should be valid for the latest version. Depending on when you install Geneious, you may be on an older version, you can update. But I'll be using the latest version for today's demo. Geneious does run on Windows, Mac, and Linux. The license is valid for all of those operating systems, so you can use whichever operating system you work with. And in addition to everything that's available within Geneious, there's also a wide variety of plugins that are available. And these are everything from plugins written by our development team through to plugins that wrap well-known third-party tools. So things like when we're talking about alignment, there's tools like Cluster, Omega, and MAFT. For tree building, there's things like FileMill and Sybase. For NGS, there's things like Fly and Minimap2 and Spades. They're all well-known, peer-reviewed tools, but they're available as plugins for Geneious. Through to plugins written by people like yourselves. So you can actually create your own plugins for Geneious. A couple of things I did want to mention about the plugins. One is they're all free to use. So there's no additional costs or charges for the plugins. The second is they're all safe to use. They've all been tested by our development team. You know, they do what they say they do. So I highly recommend, as you're taking a look at Geneious, also take a look at the plugins, because there's some interesting tools in there, and I'll show you where to find more about them. And then Geneious is supported by scientists and developers. We have a pretty amazing support team. I'm a little biased. I started out with our support team, in fact, but they all have PhDs. They're all scientists. So they can help not just with the technical questions, but with the scientific questions as well. Like I said, the support email address is in the chat. I'll also show you how you can send an email to our support team from within Geneious. And then finally, Geneious is based on the needs and expertise of people like yourselves. What that means, practically speaking, is that we really care about your feedback about Geneious. So it's always nice to hear the stuff you love using about Geneious, all the good stuff. But if you've got suggestions, features you'd like to see, comments, suggestions for improvements, definitely send that through. Probably to the support team is the best place. Anything you send through to myself or to the support team, all of that feeds back to the development team. They keep track of all of that, and that's what they look at when they look at how to grow and how to improve Geneious. So I think that's enough of slides. Let's get out of there. Like I said, the rest of this is going to be a live demo. If you do have Geneious installed, you're welcome to sort of open it up and look at where I'm clicking, but that's definitely not needed. And as I said, this is being recorded, so you can refer to the recording later. But we're going to start out with just kind of where things are in Geneious, how it works. I'll give you kind of an overview of things. And then we'll start talking through some of those tools like annotation and NGS and primers and that. But send through questions at any point. So with that, this is Geneious. Like I said, it does run on Windows, Mac, and Linux. I'm on a Mac at the moment, but it is going to look pretty much the same if you're working on Windows or Linux. There's a sort of few minor cosmetic differences. I'll point those out if we run into them. But Geneious, like I said, it's a desktop program. This is all running locally. My data is stored locally. On the left-hand side is the sources panel. At the top of this is the local folder. So this is all of my data stored within Geneious. It's specific to me. I can organize this, structure this however I like. Importing data into Geneious is pretty straightforward. Just drag and drop. You can import one file or multiple files. You can drop them either into this documents table or onto one of these folders on the left-hand side. Now these particular files are GenBank files. So they contain sequence information, but also have annotation information. So if I start zooming in, we'll start seeing all the details of the annotations. If we zoom in far enough, these are yeast chromosomes. So if we zoom in far enough, you'll see the bases as well. So at the moment, I'm in the local folder. Below this is the shared database. This is a SQL-based database. You can set up sort of within your organization, within your lab. It allows you to share data from within Geneious. So it means everybody's working from the same version of a file. It's great if you want to set up maybe a library of primers or annotations within your lab group. And then below this are some live services you can interact with from within Geneious. So things like NCBI UniProt. You can search against these. It's going to download any results. Depending on how my internet connection is going, it will eventually download some sequences. I'm searching NCBI at the moment. So this is going to be everything from DNA sequences to protein sequences. There's probably going to be some PubMed articles in here as well. Now, if you do want to keep any of these sequences, select the ones you want to keep, and then just drag and drop either to this local folder or to that shared database. So you notice when I'm clicking on these folders on the left, contents are appearing on the right. This is the documents table. To the left of each document icon, there's a small, to the left of each document name, sorry, there's a small icon that tells you what the document type is. So you can see here, I've got things, this is the red helix, that's a linear DNA sequence. Green circles, a circular DNA sequence. You can see as I click on these, I've got all sorts of things here. And when I click on them, I'm seeing the document below. So for example, so click on this one. This is a 3D structure. Geneious can visualize these. So if you import a PDB file, it can view them, but it's not going to create them. So it can't sort of de novo generate this structure. But when I click on this, You'll notice there's some tabs across the middle of my document. These are in the middle of my screen on a Mac. If you're on Windows, they're going to be slightly offset to the left. But these are different ways of thinking about this particular document. So was that a question coming through, or someone's accidentally unmuted? Okay.
Okay, I'm muting that user. Please continue.
So as I was saying, these doc tabs here are different ways of thinking about kind of the data within this document. So for my 3D structure, I can think about it in terms of the DNA sequence underlying that, or the protein sequence. I can also think about it in terms of a table of the annotations on the sequence. There's things like an info tab. If I jump to my vector sequence here, I've now got a sequence view. I've got a table of annotations again in my info tab. So we're going to talk about the sequence view for a moment, sort of how to work within that. Before I do that too much though, I do want to highlight 3 buttons on the right-hand side in the middle of my screen. If you're on an older version of Geneious, if you've had Geneious for a couple of years now, you might see 5 buttons here. It's the 3 in the middle we're talking about. So the first of these buttons allows me to split my screen. So if you want to see 2 of those tabs side by side, this is the button you're looking for. So you can see maybe your sequence view side by side with your annotations table. Alternatively, if you want to hide that documents table and sources panel, the button in the middle will help. So this allows you to hide those 2 fields, gives you a bit more room to work. If you click on that middle button again, that brings those back. And then the last button will pop your sequence out to a new window. So if you want to move the sequence to a second monitor, or you want to look at 2 sequences side by side, that's a great way to do that. So speaking of those buttons, I'm going to use the one in the middle, and we're going to hide the documents table and sources panel for now. Now on the right-hand side, I've got a control panel. This controls what my sequence looks like. I can do some basic analyses from here as well. So the very top are the zoom controls. First couple of these, pretty straightforward, zoom in, zoom out. Personally, I find myself using the 3 on the right all the time. So the 2 arrows pointing in allows me to zoom right into whatever's currently selected. The magnifying glass with an A allows you to zoom into 100%. And the 4 arrows pointing outwards allows me to zoom right back out again. So if it feels like I'm jumping around the sequences really quickly, it's usually because I'm using those 3 buttons. Now at the moment I'm in the general tab. From here I can sort of control the general layout of my sequences, so I can do things like change the color of the bases. There's all kinds of coloring schemes here. Some of these color by different information like GC content, or coloring by translation or annotation. Some of these are just for fun. Don't think anyone's found a practical purpose for this one yet. But these are all customizable as well. So if you don't see your favorite coloring scheme here, or the coloring schemes don't work for you, there's an edit button to the right, so you can change these to colors which work. From here I can turn on translation. By default, Geneious is going to translate any CDS annotations, but it's also doing this translation live. So if you highlight a region of sequence, it's going to translate that region for you. And by the way, if you click and move left to right, that's translating in the forward frame. If you click and move right to left, that's translating in the reverse frame, so on the complement strand. I'll come back to translation in just a moment. The other buttons I wanted to point out though is the circular overview. So towards the bottom of here, you can control just how your sequence is laid out. So I can show the map of my sequence maybe side by side with the sequence. And turn that off for a moment. We're going to zoom right back in again. And let's jump down to the display tab. So this is where you've got more control over the sequence itself. So I can turn on things like the complement strand. I've got more control over the translation. So instead of translating by CDS, I can translate maybe in all frames, or various frames, pretty much any combination of frames here. You can also from here change the genetic code it's using. You can also change the coloring scheme. So for example, this coloring scheme is based on different properties. You can also choose a coloring scheme that just highlights the stop codon, say. So let's jump back. I'm going to turn off translation for a moment. Actually, one other thing I'll highlight here is you can hide the nucleotides. So you can see just the translation. Just keep in mind, this is still a DNA sequence. At heart, this translation is temporary. So if you're doing anything with this DNA sequence, it's going to work like a DNA sequence. Jump down here to the graphs tab so I can show things like GC content. I zoom out, you can see that across my whole sequence. If we jump down to this yellow arrow, this is the annotations and tracks tab. So from here, you can control what annotations are visible on the sequence. So you can hide annotations. You're not losing anything. You can bring these back at any point. You can also search within here to highlight annotations. We'll jump down this orange arrow with the yellow question mark. I'm going to circle back to this in a moment, but this is a great place if you're looking for things like transferring annotations or finding open reading frames. Like I said, I'll come back to that. The button I really want to highlight though is this very last one. It's the statistics tab. This is going to give you some basic statistics on your sequence. So things like base frequency, GC content. It's also giving you statistics on the translation. So if you want to see things like the isoelectric point or the extinction coefficient, you can find that here. And again, this is all happening live. So if you highlight a region of sequence, it's going to give you the statistics for that region. So if I click on something like a CDS annotation, this is the statistics for that gene and for its translation. Now, I want to highlight a couple of last buttons here across the middle of my screen. If this wasn't expanded, they'd be right here. Now I'm working with sort of a large sequence at the moment. If I want to pull out just a piece of the sequence, I can use the extract button. So this will create a new document from whatever I had selected. Alternatively, one of the other buttons here is RC. That stands for reverse complement. So if the sequence is in the wrong direction at the moment, RC will reverse complement that sequence. And then the next button here is translate. So the translation I was talking about before on this right-hand side, that was all temporary. It was just being overlaid over the top of my sequence. Now, if you actually want to create a protein document in Geneious, the button you're looking for is this one. So translate. Again, you can do it based on the whole sequence or whatever you've currently got selected. I click translate, it creates a protein document. So it creates a new document here. It's got a little protein icon. Now, I don't need to save this document. Next time I reopen Geneious, this document's automatically there. If I do make any changes to the sequence, I can turn on allow editing and I can start typing into here. You'll notice Geneious is then going to ask me to save. So I can't move to anywhere else within Geneious without me making a decision about whether to save this or not. And then to run any analyses in Geneious, the way it generally works is that you select the sequences or the data you want to work with, and then you select the operation. And these are all across the top. So for example, I could select my sequence and go design primers, or I could do cloning. You'll notice some options are grayed out, like my tree button's grayed out. That's because you can't make a tree on one sequence. You'll notice the minute I pick 3 sequences, that's the minimum you need to make a meaningful tree. That's when the tree button becomes enabled. So the last things I want to point out just in this general kind of overview is how to get data out of Geneious, how to find plugins as well as how to find support. And then we'll start talking about things like annotations and primers. But like I said, getting data out of Geneious first, you can go to File and Export. And that's going to allow you to export your data out of Geneious. So if I do selected documents, Geneious does export to a pretty wide variety of file formats, but it's only going to offer you file formats that make sense for what you're currently working with. So I've got a DNA sequence with some annotations on it. So it's offering me things like FASTA files, GenBank, GFF. It's not going to tell me about Newick. You know, it does support it, but that file format's more for phylogenetic trees. So it just doesn't make sense at the moment. This is actually one case where Windows and Mac again look a little bit different. In Mac, it's 2 windows. If you're on Windows, it's going to go straight to something that looks like this, and there'll be a drop-down box. That's where you choose the file format. And then depending what file format you chose, you might see some additional windows that can let you specify how to format that file. Now, if we are talking about GenBank files in particular, another nice trick is you can actually just drag and drop straight to the desktop. This is in newer versions of Geneious that's going to automatically export to a GenBank file. Now, if you want to export out an image, if you want to put this in a publication or you want to put it in a presentation, to do that, go to File and Save as Image File. So you can export out to various file formats. Some of these are vector-based file formats, which means you can edit them if you resize them. It keeps that resolution. One of these in particular, you can export out and you can actually bring it into PowerPoint on Windows. If you right-click and do Ungroup, you can edit the file format within there, within PowerPoint, so you don't even need access to fancy graphics software. I believe it's the EPS file format, but I might be wrong there. It is mentioned on our support site exactly which one that is. And in general, you want to make your sequence look like you want it to look in the sequence view. So if you want to be zoomed in, do that. If you want to change colors of annotations or hide things, do all of that, and then go to File and Save as Image File. Like I said, the other 2 things I want to briefly mention, because I know we're going to talk about these a little bit, plugins. Like I said, there's heaps of things you can do within Geneious, but there's also all sorts of plugins. Easiest way to get to those is go to Tools and Plugins. This is the list of all of the available plugins for Geneious. Like I said, they're all free, they're all safe to use. So I highly recommend taking a look at these. If you click on more plugins at geneious.com, that will take you to our website. It takes you to this page here. And this is a slightly more user-friendly and more readable list of all of those plugins. Some of these are also open source. So if you're interested in developing your own plugins, there's some great examples here of how to do that. And the other thing I wanted to mention now is just how to get more support on Geneious. There is a Contact Us button in Geneious. So you can send an email. This will send an email to our support site, our support team. You can also click on this link to Knowledge Base. That will take you to our support site, which looks like this. From here, you'll find all sorts of frequently asked questions. You'll find a link to the Geneious user manual. The manual is more like an encyclopedia. It's a great place to go to if you're wondering how certain algorithms work in Geneious or what it's doing. If you're looking for more of a how-to guide, then I highly recommend the tutorials. These will walk you through various analyses in Geneious using real data. So you can import them into Geneious. So for example, there's a tutorial here on how to create workflows. Import it into Geneious and it'll show you how to work through that. Just want to stop at this point. Are there any questions from here? I'm going to talk a little bit about, let's go with annotations. We'll also do some simple alignments and tree building and talk about the various alignment functions in Geneious, as well as talking about primers and NGS. But are there any questions at this point? Anything I might have forgotten to cover that anyone wants to raise or make sure that I cover later? Okay, don't forget to unmute yourselves if you are asking questions. I'll keep an eye on that chat function as well in case anything's coming through. Hopefully that's a good kind of silence. So let's jump straight into annotations. We're going to talk about, again, this is one of my other favorite tools within Geneious. So I've got a sequence here. It's an empty sequence I've imported into Geneious. And one of the tabs I skipped over earlier was this orange arrow with a yellow question mark. It's the Live, Annotate, and Predict tab. So from here, I can do things like find where any open reading frames are. I can control the size of those open reading frames. So you can see that changes what it finds. Now, if I like these, the results here are all temporary. So if I turn this off, they disappear. If I want to keep these, I need to click the Apply button. So that's actually going to add the annotations to the sequence. Now, in this case, I've applied all of my annotations. Alternatively, I can just select one or some of them and click the Apply button, and that's going to add just that particular annotation. So that's one way to find open reading frames in Geneious. Be aware it is sort of a simple or naive open reading frame finder. So what I mean is it looks for a start codon, and then it keeps going until it finds a stop codon. So it's not handling things like introns and exons. There are two plugins for this, Glimmer and Augustus are available as plugins. They can also be used for finding open reading frames. But the tool above this is one of my favorites. It's called Annotate From. Now this tool can be used to find annotations in your sequence. So I'm going to drop this down a little bit. You can see as I'm changing the similarity, it's changing what annotations it's finding. Now again, everything here is temporary. If I turn this off, they disappear. If I want to keep these annotations, I need to click the Apply button. Now the obvious question here is, where are these annotations coming from? What's going on? Now in this version of Geneious and the latest versions of Geneious, they're coming from a place called reference features. If you're using a slightly older version of Geneious, you'll notice it's pointing to the plasmapper features, which is in the sample documents. But like I said, I'm in the latest version of Geneious, so it's pointing to my reference features folder, which is this folder down here. So this is a curated library of common plasmid features. It's actually a mixture of DNA sequences. There's also some protein sequences in here. So this is a curated read-only library, but Geneious is actually pointing to this entire folder, and this folder is editable. So you can add your own sequences in here, so you don't need to just use the plasmid features. You can add and create your own sequences. So to give you an example of that, I've got two sequences. One is the mitochondrial genome from an emu, which is this large flightless bird from Australia. And the second is the mitochondrial genome from a kiwi. Now, I don't mean the fruit, I mean the bird. It's this small, much cuter flightless bird from New Zealand. Now, I'm a little bit biased if you've been spending this whole time just trying to guess where my accent is from. It's a New Zealand accent. And in fact, that's where our development team is based. That's where the support team is based as well. They're all working out of New Zealand. But the emu and the kiwi are closely enough related that I can use the emu to annotate the kiwi sequence. And it's really simple. I'm just going to grab this emu sequence and I can put it directly in this reference features folder, or I can create my own folder within here. I can move that emu sequence to there. So now when I jump to that kiwi sequence and turn on this tool, it's automatically searching against any annotation within this reference features folder. Alternatively, instead of wasting time having a look at all the plasmid features, I can click on this and ask it to search against specifically the emu folder, or actually any other folder anywhere in my Geneious database. So click OK. Again, everything's temporary. If I turn this off, they disappear. If I click the Apply button, that adds the annotations to the sequence. Like I said before, this will actually work from DNA to DNA, or protein to DNA, or protein to protein. So if you have something which has been codon optimized, for example, or just very divergent, then you can potentially put protein sequences in there and still successfully annotate your DNA sequences. So that's kind of how annotations work in Geneious. Any questions at this point in specifically about this tool or generally about annotations in Geneious? I think from here I'll move on and talk maybe a little bit about, let's talk a little bit about primers next. But any questions at this point? Anything anyone's wondering about annotations? Okay, hopefully I'm not about to speak over anybody. Do speak up if you're still trying to find the unmute button. Let's talk next a little bit about primers in Geneious. Like I said, we're jumping around topics a little bit. If I don't manage to cover what you're interested in, there are tutorials on all of this kind of thing on the website as well. But let's talk a little bit about primers in Geneious. So I've got a sequence here. We're going to design some primers on it. Now I'm going to clean up the view. You can design primers directly on this, but for personal preference, at the moment, I only care about my primers relative to my CDS annotation, say. So I'm going to clean up this view so that I can see just the CDS annotation. I'm also going to turn off wrap. Again, kind of personal preference. Personally, I find this a little bit easier when looking for pairs of primers, but you can change this however works for you. So design primers in Geneious, that's all under the primers menu. I'm going to click on the CDS annotation and go to design new primers. So the first set of options up here control where the primers are going to bind to, where they'll be targeted to. And with the default options, these are really generalized where it's going to bind to. So if I turn on included region, you'll notice Geneious is pre-filling with the range I'd selected. So I'd selected my CDS. It's pre-filled this range with the coordinates for my CDS. So if I do included region, it's going to design pairs of primers that are inside of my CDS. They may be anywhere within that, but they're certainly going to capture some of the CDS. If I do target region, Geneious is going to design primers that are outside of my CDS. Again, they may be anywhere upstream and downstream of that CDS, but they'll certainly capture the whole CDS, but might capture some additional information. And you can use both of these options, so nest them. So I could say I want primers that are within 100 base pairs upstream and a primer that's within 100 bases downstream, say. So if I click OK, and I've got that round the wrong way. I have. Let me just change that round. All right, so if I do it this way, it's going to contain, in this case, create primers that are within the 100 base pairs either side of my CDS annotation. Now, if I hold my mouse over this primer, a tooltip appears. That tells me a little bit more information about the primer. So things like GC content, melting temperatures for any hairpins, self-dimers. In later versions of Geneious as well, it's also going to tell you if these primers bind elsewhere within this sequence, and a bit more information as well. Now, if I don't like these primers, if I've changed my mind about them or I got the settings wrong, getting rid of them is as simple as click on the primer annotation and hit the delete key. So hitting the delete key on your keyboard, that deletes the primers. Alternatively, I can jump to this yellow arrow, the Annotations and Tracks tab on the right-hand side. If I click on the green line next to primers, I can delete all of these primers at once. Now, if you're doing primer design for cloning, just be aware when I go back into design new primers, instead of keeping my task on generic, I can change this to precise. I only need one pair of primers. This is going to design primers that are anchored to my current region. So one primer starts at the beginning of my CDS, one primer begins at the end of my CDS. Now, I'm not going to go too much into primers at the moment, just in the interest of time, but just a few other things to be aware of. When you're doing primer design, you may run into this message occasionally. This is because Geneious not only is trying to find primers, but it's trying to find primers that satisfy all these criteria. So if I go back into primers and design new primers, it's trying to find primers which meet all these characteristics. So my primer needs to be within a certain size range. Its melting temperature needs to be within this range. Its GC content needs to be within this range. And depending on how narrow the region that you're trying to design your primers within, or just the sequence itself, it may not be able to find primers that match those criteria. So you can edit these. I can say, I don't mind what my GC content is. I don't mind if my primer is a little bit smaller, a little bit larger. You can change all of this. It may be able to find primers. Alternatively, you can always design them by eye. So if I zoom in, I can highlight a region of sequence. It's going to tell me what the rough temperature is. If I have that statistics tab open, I can also see things like what the GC content is. And I can say to myself, this looks pretty good. When I let go of my mouse, green arrow appears. I can add a primer directly to the sequence. So now this is a primer like any other primer annotation in Geneious. The other thing I want to mention is you can add 5' extensions to your primers. So if I double click on this, I can add restriction sites or bases onto my primers. Click OK. We're just going to add these onto my primer. You can see that it gets added to the end of the sequence. You can also do that, by the way, from within the primers options, design new primers. At the very bottom, there's some advanced settings. This will also allow you to add extensions directly onto the primers it creates. So that's a little bit about how to create primers in Geneious. There's multiple places you can go from here. You can go straight to extract PCR product. That's under the primers menu. If we jump to the annotations tab, you can see all of my primers. I'm going to filter this by my primers, and I can actually export these out of here as a CSV file if I want to order them. If I want to create primer documents within Geneious, I can click on that primer annotation and click extract. And that's going to create a new document in my Geneious database. So now when I jump to another sequence, I could do tests with saved primers and see if I've got any primers, existing primers, anywhere else in Geneious that would work with this sequence. And you can see I don't need to store them in a specific folder in Geneious. Geneious kind of just knows where my primer documents are. So I can say, look, anywhere in Geneious, or go into this folder, see if I've got a primer that would work. And if I do, it will annotate it back onto my sequence. The last thing I want to mention about primers, we've talked about doing primers for creating sequences, creating PCR products. Just be aware, another option under here is there's an option called Design for Sequencing. So if you're doing something like Sanger sequencing, you can ask Geneious to create primers which are going to tile across your sequence, so be spaced at like 500 base pair intervals, and it will create those that sit across your sequence. The last thing I'll mention on primers is just kind of a general tip for Geneious, but it is commercial software, but we try to be very open about how we're doing things and what we're doing. So you'll notice in a lot of places in Geneious, it'll tell you what algorithms it's using, or it'll tell you a citation. So in the case of primer design, Geneious is using primer 3. So if you're familiar with primer 3, this might actually look familiar to you. But it's using specifically version 2.3.7. And there's a link here. So you can go learn more, read up about how primer 3 works, what it does. So that's a little bit about primers. You can also do designing primers on things like annotation, on alignments. The next thing I think we're going to talk a little bit about is the various alignment options in Geneious. Any questions though on primers, or anything else on anyone's mind at the moment you want to bring up? Okay, so there is a question that's come through the chat. Does Geneious contain most of the commercial cloning or expression vectors, or do we have to upload them from the source? You probably do have to upload them from the source. You'll notice there are some things in the sample documents, so things like plasmids from NEB, but this is not updated. So there are some within Geneious, these specifically coming from NEB, but you can also upload them from various sources. If you find them on a source in something, usually a GenBank file format is the best one to look for, then you'll be able to import that into Geneious. If you're looking at something like SnapGen has a pretty good curated library of vectors, you can import those in either as a SnapGen file or a GenBank file straight into Geneious as well. Hopefully that helps answer your question. Any additional questions on that? Let me know if that didn't cover it, or any other questions from anyone. Okay. So I'm not going to cover cloning at the moment, unless we might have some time at the end if there's requests for it. There are some tutorials on the website. Instead, we're going to jump a little bit and talk about alignments in general in Geneious. We're going to talk a little bit about NGS. We'll talk about just multiple sequence alignments. We can maybe cover some Sanger sequencing. I'll show you a quick taste of workflows as well, because they tie particularly nicely into this. But what I first wanted to cover, I guess, is what the alignment options are in Geneious. They're all under this Align Assemble button, because there's a few different options here. They all do kind of similar things, and I know the word align sort of gets used around in the literature with different meanings, different programs use it in different ways. So I guess before we get too deep into this, I want to talk a little bit about what these different options do. And to give you an example of this, I've got a sequence. So in this case, this is the mitochondrial genome. I think this is from a lion. So I've got that mitochondrial genome here. Now, if I took one gene out of this sequence, maybe let's extract out this particular sequence, and I had the same gene from other species, so same gene but from other vertebrate species, and I want to align them together, that would be what's called multiple sequence alignment. So all of these sequences I've got here from the same gene, roughly the same length, they're roughly expected to align against each other. So this sequence is expected to be similar with this sequence, which is similar to this one, which is similar to this one. So if we're talking about this, that's when you would use multiple align. And these could be DNA or protein sequences. So I'm going to come back to this, talk a bit about multiple align in a moment. Alternatively, if I've got, again, my mitochondrial genome, maybe I've sequenced this either with Sanger sequencing or NGS. So I've got a sample from a single lion. I've done NGS sequencing on the mitochondrial genome. So I've got data that looks like this. Lots of sequencing reads. These all come from the same sample, but they're just small pieces of a larger picture. So this particular sequencing read here isn't expected to have anything in common with this one or this one, say, but they are all from the same sample. That's when you would use map to reference or de novo assembly. These particular tools are designed to work with Sanger sequencing or NGS data. The advantage of them is they understand that your reads may have contamination in them, so they'll throw out anything that's too dissimilar from your reference or too dissimilar from each other. They also understand things might be sequenced in different directions, so they're going to automatically flip sequences if they need to. Most of the multiple alignment tools assume everything's in the same direction, assumes that everything should be aligned together, so it's going to align it regardless of whether it should be aligned. Map to reference de novo assembly assumes that's not the case. So you'll end up with something like this if we've got a reference sequence. So all of these individual reads, here are just one small piece of that larger picture. And then the final option, or final case you might find yourself in, is where I've got, again, that mitochondrial genome, but this time I've got the mitochondrial genome from other species as well. So it's a much larger piece of sequence, and whilst they're generally expected to be similar, maybe there's rearrangements. Maybe these genes aren't in the same order across all these species. And that's when you want to use something like align whole genomes. In particular, there's two plugins here. You can use Mauve or Mauve, if you've got the American accent. Mauve is going to allow you to align multiple genomes, or there's a plugin called last Z, that's going to allow you to do a pairwise comparison of those sequences. So this is giving you an alignment again, but it's saying, hey, this region of this sequence, it's actually found over here on this one and over here on this sequence. So that's what align whole genomes is perfect for. So we're going to talk through a few of these options now. We're going to start out with the first one, multiple alignment, where we've got multiple sequences from the same genome, from the same gene, but from across different samples or across different species. So I'm going to go to align assemble and multiple align. So you can see there's some different algorithms, different methods I can choose from here. It's as simple as just click the method you want to work with. There's no right or best method here per se. You'll get different results with different methods with different data sets. The Geneious alignment method works great up until like about 50 sequences. After that, other methods are going to be a lot faster. MAFT is a plugin, so you'll need to install that plugin first from that plugins menu, but it's really great with large data sets. So if you've got thousands of sequences, MAFT can handle that. I also personally like this method because it also allows you to determine the sequence direction. So if you do have one or two sequences that might be in the wrong direction, it can handle that. So we'll click OK. This is going to run through, creates my multiple sequence alignment, gives me this. So you can see anything like annotations, which I had, still visible here, and I can turn them off if I don't want them. And turn off my translation as well. Now, by default, Geneious has highlighting turned on. So it's highlighting any disagreements to my consensus sequence. And that's all controlled from the display tab on the right-hand side. Now, by default, these kind of alignments, multiple alignments, don't have a reference sequence. It just aligns everything against everything. But you can choose a reference sequence after the fact. So in this case, let's go with lion. Let's make that my reference sequence. I can right-click on it and do set as reference sequence. And you'll see my highlighting automatically updates. So it's now highlighting relative to that reference sequence. The line sequence has a yellow background. I can actually click and drag on it, move it to the top of my sequence, top of my alignment. I can click and drag any of these sequences this way. If you right click on one of these sequence names, you can actually sort the sequences within the alignment. So I can, for example, sort by their number of disagreements to that reference sequence. So if we zoom in, we can see all of the details within this alignment. So you can turn highlighting on or off. One thing I particularly like to do is you can turn on use dots as well. That makes it really easy to spot where those disagreements are. And all of those other options we've talked about before, everything like translation, all still applies here. So I can translate. I'm going to translate based on frame 1. You can see even highlighting applies to that translation. I can hide things like the nucleotides. If I jump to the statistics tab, it's going to give me stats on this alignment as well. And at the moment, I think my translation is using the wrong genetic code, so I can change all of that as well. So if I change this to the vertebrate mitochondrial code, it's yes, doing got rid of all those stop codons that shouldn't be there. Now, one thing to be aware of here is this is still a DNA alignment. I aligned it as DNA sequences, which means it hasn't necessarily put gaps in the best of places. So the gaps aren't necessarily in groups of 3. They might be in the middle of codons. So if we scroll down here, is a good example. So these gaps aren't necessarily in places that make sense. So if you are working with coding sequences or protein sequences, I can realign these sequences. And there's an option here called translation align. So what this is going to do is translate all of those DNA sequences according to the genetic code I give it, align them as protein sequences, and then back translate everything. So I'm still going to get a DNA sequence alignment. Bring back my nucleotides there. It's still a DNA sequence alignment, but now my gaps are in sensible positions that are respecting where my codon boundaries are. Alternatively, you can use that translate button. I can translate all of these sequences. So now they're actually protein sequences, and then the exact same button. So align, assemble, multiple align. I can do a protein sequence alignment using the same options the same way. This is going to run through, it's going to do my alignment. Like I said, some of the other alignment algorithms are a little bit faster than the Geneious one. And this is what a protein alignment looks like. So it's going to default to a coloring scheme called similarity. Like I said, you can change back to any of the other coloring schemes, whatever works best for you. Again, turn on highlighting, all of that's available here. All of the statistics are going to be for the protein alignment. So this is multiple sequence alignment with homologous sequences, pieces that are from the same gene or the same region of DNA. If you're doing pairwise, exact same process. So I can pick two sequences here. Same place even, that button is going to change its name. It's going to go from multiple align to pairwise align. So I know one of the things people had asked about was building phylogenetic trees. So I'm going to talk about that next before we circle back to things like NGS. But any questions at this point about multiple sequence alignments or anything else that sort of come to mind? Okay. Like I said, hopefully I'm not cutting anybody off. Do speak up if you're struggling to find that mute button there, or send it through the chat function. But let's talk about trees then. So I've got a multiple sequence alignment here. If I want to build a tree from this, it's as simple as click the tree button. So if I click tree, by default, the inbuilt tree building method in Geneious is neighbor joining or UPGMA. These are distance-based tree building methods. They're really great. They're quick and dirty tree building methods. They'll give you an answer very fast. They work great with really large data sets. They're distance-based tree building methods. So they work well, but they don't work well in situations where you've got very different mutation rates, or if you want to find sort of more the evolutionary history, or if you want to put this in a publication, they're not necessarily the best tree building methods. There are plugins for things like FIML and Mr. Bayes, Gali and RexML. You can use those as plugins from within Geneious, and if you have them installed, they're going to appear right here. But I'm going to keep this simple. We'll just use neighbor joining tree building method. What I am going to do is turn on resampling, and I'll show you why that's important in a moment, but we'll turn on resampling. Click OK. Geneious builds a tree. So this is my tree building, my tree window, very similar to my sequence window. On the right-hand side, there's a control panel. So I can change things from here. One of my favorite things, and what I wish I'd had when I was studying trees, is at the very top, there's a search window. So if you're looking for particular sequences, it doesn't take a lot of sequences in a tree before they're hard to spot, you can search from them here. Now, one of the things I did when I built this tree was I turned on resampling. That's going to give me confidence values on my tree. Now, it's very hard when you're looking at this tree like this to say how well my data actually supported this tree. You know, does the sequence alignment strongly support this grouping of sequences, or could it have equally as likely been something else grouped differently? What about this one? So that's why I turned on resampling. It gives me a confidence value. So if I turn on show branch labels, you can see that's what these numbers at the clades, the roots of my tree are. So this is saying, so this particular grouping, this had 100%. While some of these groupings here are much lower support within the alignment for that grouping of sequences. So one of the other things I want to highlight here is those different tabs that are available here. So I can see my sequences that were aligned, but now they're sorted relative to that tree. So you can see here they're aligned relative to that tree structure. I've also got a distances tab. This is a table of all of the distances between my sequences. So I can do it based on branch lengths or things like percent identity. I can turn on heat map here as well. So that's trees in Geneious. What I would say here, just a general piece of advice, is if you are going to work with trees, you definitely want to build the alignment first. So do the multiple line and then build the tree. You may notice that if you click sequences, kind of like I had before, these are unaligned sequences. I can actually go directly to tree from here. I don't recommend doing this. What's going to happen if you do it this way is it's going to create pairwise alignments of all of these sequences. So it'll align this one to this one, and this one to this one, create pairwise alignments. It's going to then condense that information down into a single number of distance, how far apart that pair is, creates a large matrix, an array of all of those numbers, and then it builds a tree on those numbers. The problem with doing this approach, if you're going straight from unaligned sequences to a tree, is that you don't get to see what those pairwise alignments look like. You don't get to see what that alignment that generated that number is. So it's impossible to tell when you get to this point if the weird grouping you're seeing or the weird clusterings you're seeing are because it's genuine data, genuine interest in your sequences, or it's because those pairwise alignments went wrong somewhere. So I would highly recommend create the multiple sequence alignment first, then go do the tree. Don't try skipping that step. With that, any questions at this point? Anything on anyone's mind? I think what I will show you very quickly is a workflow, just because this lends itself to a very nice simple workflow. Give you an idea of how those work. And then we'll start talking a little bit about NGS data. Like I said, workflows my absolute favorite tool within Geneious. Certainly don't start out with workflows straight away, but once you've kind of got some familiarity with Geneious, you've got an idea of what you're doing within the software. If you find there's certain things you're doing again and again, absolutely go take a look at workflows. So workflows can be as simple or as complex as you like. So what we've just been doing, that lends itself to a very nice simple workflow. You can have a workflow that just builds that alignment, builds that tree for you. So it's still a case of a one button click, but it is doing that alignment and you can go verify that alignment. You can get kind of as complex as you like with these. This is one of the fancier workflows. This particular workflow does de novo assembly and reference mapping, and it picks up documents from different steps. So you can make these as complex as you like. But they're relatively simple to create. Just go to new workflow and you add those steps piece by step piece. So I can create my alignment using math, say, and then use a tree. I could pick any of these tree building methods. I can double click on any of these steps in the workflow and change all of the settings. So I can preset them so that I never need to care about them or worry about them when I run the workflow, or I can expose some or all of them so that I can change particular settings each time I run that workflow. What I would say is build the workflow step by step. Don't create a workflow with 10 steps, run it, and then hope that it works. Instead, put your first step in there, run that, check that the results are what you expect them to be, then go back, add second step, run that, check that the results are what you think they should be, and go from there. Like I said, there's some tutorial on our website that talks more about how to create workflows. You're also welcome to reach out to our support team. The other really cool thing you can do from within workflows is you can add custom Java code. So if you're feeling a bit adventurous or you want to add something new into Geneious, that's a really great way to do that. Any other questions? Any questions at this point? If there's no questions, the next thing we're going to talk about is NGS data. Like I said, Geneious works with NGS or works with Sanger sequencing. We're going to go through an example with some NGS data. A lot of what we're doing though would actually apply to Sanger sequencing. So if you're working with Sanger data, you'll still get a general idea from this. Now, Geneious can do the pre-processing, so I can bring in straight from a FASTQ file. It can do the pre-processing of my data. It can map it to a reference sequence or de novo assembly, and then it can call my SNPs. That being said, you can jump into that pipeline or into that process at any point. So if your sequencing provider has already done some pre-processing, skip over those steps in Geneious. If you've already aligned your sequences somewhere else, so you've got a BAM file instead, then just import that BAM file straight into Geneious. You don't need to rerun or redo work necessarily within Geneious. So you could go straight from a BAM file, which gives you a config, and then you can go straight from the visualization and do your snip calling. But for the purposes of a demo, I'm going to go straight sort of from the beginning steps, and we'll walk through the whole process. So I'm going to start with some FastQ files. And I'm going to import these into Geneious. So I've got 2 here, just drag and drop them in like I did with those other sequences. Now for FastQ files specifically, options are going to appear. Geneious is going to ask you what the read technology is. So is this Illumina data? Is it nanopore data? Tell Geneious what this is. What it's going to do is keep a sort of note in the back of its mind. So when it does things like de novo assembly or reference mapping, it's going to be able to tweak the settings internally to better handle that data. So better handle how that data has errors. This is Illumina, so I'm going to tell it that. Then if your data is paired, tell Geneious this, as well as tell Geneious what direction, what orientation the pairs are expected in, as well as how far apart what the insert size is. So starting from the beginning of one read through to the end of the other read. It does realize this is an average. Not all reads are going to be exactly this insert size, but just put the average, the expected distance here. So this is going to import in my sequences. This is all of my raw reads. You'll notice to the left of my sequence name, there's a little blue icon. So when I told Geneious it was paired data, it didn't really do anything with that. It hasn't merged reads or anything. It's just kept a note again in the back of its mind. This is paired data. When I do my assemblies, I'll take advantage of that fact and use that if it's available. So because I've got quality scores on this data, Geneious has defaulted to coloring my reads by the quality score. So dark blue is low quality bases, light blue is high quality. And if you want to see the actual quality scores associated with each of these sequences, that's appearing in the status bar at the very bottom. So you can see as I move my mouse over this, what I'm seeing in that status bar changes. And again, things like the statistics tab, they're all available here. So it's now giving me information, statistics across all of these sequences. So from here, there's all sorts of things I can do. I'm going to run a tool. I'm going to do some trimming on my sequences, and I'm going to use a tool called bbduck to do that. Now, this is one of the plugins in Geneious. So I installed it from that plugins menu, tools, plugins. I personally particularly like this tool because it's going to allow me to trim off the adapters, as well as throw out short reads and trim by quality score all in one step. So I'm going to go to annotate and predict, trim using bbduck. I'm going to ask it to trim my adapters, trim based on quality score, and throw out short reads. And it's going to run through and do that. Now, like I said, if your sequencing provider has already presented you with trimmed data, then skip over that step. If under the sequence menu, there's also additional tools like merging reads if they are overlapping, as well as removing duplicate reads and error correction. Now in this example, the next step I'm going to do is map my reads to a reference sequence. There's a good question that's come through the chat function here about the capabilities. This is desktop software and what you can and can't do here. So we're going to do some reference mapping. I'm going to select my reads and go to map to reference. Just going to reset to the default settings. If you ever want to reset to the defaults, bottom left-hand corner, there's a gear icon. That's going to allow you to reset to defaults. If it's grayed out, it means it's already on the defaults. Now, when you're working with NGS data, just in general, in Geneious or elsewhere, it can require a lot of computer power, it can require a lot of RAM, and Geneious is a desktop computer, desktop program. In general, de novo assembly is going to require the most power in Geneious. So if I do de novo assembly, this is sort of the most computer spec intensive tools. Map to reference is not as greedy. But Geneious is going to give you an estimate of how much it's going to need. So you can see for this particular reference mapping, it only wants 84 megabytes. I've actually got 13 gigabytes available. For de novo assembly, it might require a lot more. For de novo assembly, a good rule of thumb is like 1 gigabyte per 1 million Illumina reads. but it really depends what setting you're using, what kind of data you're using, which de novo assembly algorithm you're using as to how much specs it's actually going to need. So Geneious for NGS data is going to work great for mapping to a reference. For de novo assembly, it's going to be able to de novo assemble things like viral genomes, bacterial genomes will probably work well. The Geneious de novo assembler is not going to de novo assemble the human genome. It's not going to de novo assemble large eukaryote genomes. Some of the plugins may, but you're definitely going to require a lot of RAM or something like a server for that. Like I said, map to reference doesn't require as much power. Again, it is going to depend a little bit on your computer itself though, as to how much or what you can do here. Hopefully that answers your question that came through the chat. Let me know if there's any other sort of follow-up questions to that, or anyone else has anything. I'm going to use the Geneious reference mapper at the moment. So there's a couple of things here I should point out. I'm going to use the Geneious reference mapper. Like I said, there's other reference mappers you can use. Most of them are available as plugins. If you've got the plugin installed, you can pick them from this list here. There's a question mark button to the right-hand side. It tells you a bit more information about sort of the basic high-level pros and cons of each of those methods. And then at the top, I can choose my reference sequence. So you can actually choose your reference sequence from anywhere in Geneious. So I could use those sequences I imported from NCBI before, or my GenBank files. I can choose multiple sequences. I'm going to keep this simple. We'll just use this one. And I'm going to click OK. So this is going to run through. This is a very small sample data set, so it runs very quickly. It gives me a config. So this is all of my reads mapped to that reference sequence. I'm just using a single gene example. This could be something much larger. So for example, what I had before, it could be even larger than this, but this is what it looks like when, say, it's a mitochondrial genome, and it's mapped across that. I've got my config. Let's hide that documents table in my sources panel. So by default, again, highlighting will be turned on. I can color my reads by things like paired distance. So this is, did they actually map within that expected size I gave before? If it's green, it's all good. Everything is sort of lining up with how I expected it to. If it's all yellow or blue, it means my reads are much further apart or much closer than what I was expecting, which either means I had a typo earlier or maybe something went wrong with the sequencing itself. I'm going to change back to one of these normal coloring schemes. As we start zooming in, you'll start seeing highlighting appears, just like we had with our alignments, our multiple sequence alignments. Highlighting appears, again, it's highlighting relative to that relative to the reference sequence. Everything is controlled from that display tab. If we zoom in far enough, you can see all of the details here. Now it's really easy with NGS data to spot where my differences are relative to my reference. I can spot where these SNPs are. That's not so much fun though if you're talking about lots of data sets or you've got lots of these. So one tool I want to highlight is under the Annotate and Predict menu. It's called Find Variations in SNPs. This is going to allow you to automatically annotate where all of those SNPs are. And then once that information's in a table, it means you can sort and filter by that. So we're going to go into Find Variations in SNPs. Now these settings at the top, they control what is going to count as a SNP. So you do want to think a little bit about your data, where you got your data from, and how that impacts how frequent something is going to be before it counts as a true SNP. So are your sequences haploid? Are they diploid? Are they polyploid? Is your sample from one organism or is it from multiple organisms? All of that is going to impact how frequent something is going to be here, whether it's a true SNP or not. So definitely keep an eye on these settings and change them as makes sense for your data. So I'm going to click OK. And this is annotating where all of my SNPs are. So it's added these new annotations. If I hold my mouse over them, like I did with the primer annotations before, tooltip appears and it tells me more information about that particular SNP. So what the changes, it's a C in my reference sequence, it's a here, so this is an A. How frequent it is. So there's actually 48 reads that cover this particular site. Of those, 48 have this mutation, which means it's at a frequency of 100% within this data set. And if you've got CDS annotations on here, it's also going to tell you information about how that affects the protein sequence. So in this case, it causes a codon change, but that does not cause an amino acid change. which means it has no effect on my protein. And one thing I really like to do here is I've got all of my SNPs annotated on my sequence. That button I talked about earlier, splitting my screen, really useful here. I'm going to split left and right and jump to my annotations tab. So now I can have my table of my SNPs side by side with the sequence itself. So I can start filtering these and sorting them. So I can look for all of the SNPs that cause a substitution, say. And if I click on these, I can jump directly to those SNPs. Or I can see if there's any SNPs that cause a frame shift or an extension. So I can control all of that from here. I can also export it. So a CSV or a TSV file, export that out of Geneious. That's kind of an overview of how Geneious data works. In Geneious, there's things like plugins for Freebase if you want to use that instead for calling your SNPs. Like I said, it does de novo assembly. This is another case where what we just did, you could create a workflow that does this. So I could create a workflow that trims my sequences, maps them to a reference sequence, and does that find variations in SNPs. So I could create a workflow that does all of that as well. So where we've just got 10 minutes remaining, the last thing I was going to very quickly show you, and I don't have a lot to show here, is MOVE alignments, which is what you do when you're working with whole genomes. But are there any questions on NGS data? Does anyone want to see a very quick demo of Sanger sequencing?
Hi, Helen. This is Rebecca. I had a quick question on the Find Variation in SNP tool.
Yeah.
Yeah, so what you displayed, is that a variant caller, or is it just displaying any mismatch with the reference?
So it's a little bit of both. It all comes down to how you set these 4 settings here. If you turn all 4 of them off, it is showing you any mismatch to the reference sequence. It doesn't matter how frequent it is, any mismatch. If you start turning these on and changing these settings, then it's showing you what it believes under those settings are actual variants, actual SNPs.
I see. And so there aren't, well, maybe you mentioned that Freebase is included as one of the algorithms. So these are the 2 tools you can use basically to call variants?
Yes, yeah.
Okay, thank you.
So if you install Freebase, it's under the plugins menu, just click install. I should mention where, if you install these plugins, where they end up showing up depends on what the plugin does. So if you install something like Bowtie or BBMap, they're going to appear when I do reference mapping or de novo assembly. Something like Freebase appears under the annotate and predict menu, appears right under the other SNP finder. In its case of just picking Freebase. A similar thing where it annotates these on, it creates a new track of variants on my sequence. Because it's a different algorithm, it's got some other information attached to that. It also sorts them by tracks, which means I can hide, I can keep both of them there and just choose which one I'm showing at any time. Any other questions around NGS data? Does that answer your question? Anything else at all? Or anything else, other topics, anything people want to make sure I try and cover in these last 10 minutes? So there's a question that's come through about how the track function works. All right, so the track function is kind of a way of grouping your annotations. Some things like doing variant finding in Freebase will automatically put the annotations they create into a track. So when I double click on an annotation, it's really just, I double click on an annotation, opens up this edit annotation window. The third option here is a track. And it's just a case of I can move annotations from various tracks. I can create new tracks here. So if I click OK, I've just moved that particular annotation up to being outside of a track. Same way I could click on my CDS annotation. I could choose from the existing tracks. I could even type directly in here. That's just going to create a new track. You can see it's automatically added it down here. They're really just a way of grouping your annotations. So Freebase and variants will automatically be imported or shown or organized by tracks. If you import a GFF file or a bed file, those annotations will also be imported onto tracks. And then from within here, you can sort and filter by those track types. Alternatively, like I said, you can create your own tracks and create them yourself. Does that, I don't know if I've explained that very well. Does that help answer your question, or did you have any sort of more questions on that, or anybody else? Okay, question on DSEQ2. Okay, so yes, Geneious does, like I said earlier, does allow you to do basic RNA-seq analysis. A lot of that process is very similar to what we've just done. So let me grab some data here. So if you're working with RNA-seq data and you want to do expression analysis, very similar process to what we've just done. I've got my NGS data. I can do the trimming or the pre-processing. I can grab all of my read lists and map them to a reference sequence using exact same tools. You can either use the Geneious mapper, or if it's RNA-seq data where there's splicing in that, you can use the Geneious RNA-seq mapper. This all works the same. You'll notice at this point I've selected multiple samples here. So in the very top, if I want to treat those as all independent and do this in batch, I can actually click this one button up here, assemble each sequence list separately, and it's going to do my mapping for all of these data sets independently against that reference sequence. So I don't need to do this 6 times. So that'll run through. I'm not going to make you sit and wait for that to finish. Essentially what you get is 1 contig per sample. Now, as I said, it does basic RNA-seq expression analysis. What I mean by that is it will do 2 conditions and multiple replicates, and it does that using DSEQ2. So do that, go to annotate and predict and calculate expression levels. This is going to calculate the RPKM, the FPKM, and TPM on all of my sequences. I do need to do this contig by contig. That's because if I had multiple chromosomes for my reference sequence, I would have got 1 contig per chromosome and per sample. So if I had multiple chromosomes, say this was the same sample set, but 2 different chromosomes, I'd want to calculate these in batch. Or if it's from 1 sample, I'd want to calculate this independently. That's so that it's correctly taking into account the actual read levels across the sample as a whole. Not sure I've explained that very well, but if you play around with this with your own data, hopefully that'll make a little bit more sense. And what's happening at the moment, and this is another great example of tracks at work, is every time I run this tool on each of these data sets, it's creating a new track. So it's calculating the RPKM, FPKM, and TPM, all these sequences. So here they all are, for the 4 I've done so far. If I hold my mouse over it, it gives me all of those statistics. And each time I've been doing this, it's been asking me if I want to apply them back to my reference sequence. So when I jump back to the original reference sequence, all of those contigs I created within Geneious are all linked back to this sequence, and it's been applying all of those tracks of annotation back to that sequence. So I've got them all here from across all of my samples. So now within this data set, I can go to compare expression levels. And this is where DSEQ2 comes into play. So it's just a case, choose that option. In this case, I've got metadata attached, something in that info tab that tells me what all of these samples is from. So it's pulling out that sample condition and using that. Alternatively, I can assign them manually. And click OK, and this is going to run. Now, the very first time you run it, it's going to need an internet connection so that it can download the DSEQ2 package. Any subsequent time you run it, it'll just run automatically. So it'll take a little bit longer the very first time, be much quicker the other times. What it does, creates another track. This is of the differential expression values. So this is where I find all of these statistics. And what I really like about this, there's a couple of things I should mention here, I guess. I could have always exported all of this information. So I could, if I wasn't doing 2 conditions, if I was doing more, I could have exported this whole table out, gone and analyzed it somewhere else. But what I particularly like doing about this in Geneious is because I've done that comparison within Geneious, when I save the changes, I get my PCA plot and my volcano plot. And this volcano plot is linked to my sequence view. So if I click on a dot within the volcano plot, so this outlier here, jump back to my sequence view, it selects that annotation for me. So I can zoom in, that is that sequence. What's really nice about this is So I can click on any of these for things like, you know, this one is down here, hypothetical protein. You know, there's a ton of these on my sequence. I don't need to know which hypothetical protein it is or match it up. It's this one. It'll jump straight to that sequence for me. So it's a little bit about how RNA-seq analysis works and DSeq too. Hopefully that helps. The other cool thing, useful tip I'll point out about annotations, we were talking about tracks before. The other really great thing that you may not discover but can be really useful, a couple of like minor features here, is if you double click on an annotation, we were talking about the track feature here. If we expand the properties tab, from here you can change the color of your annotations. So all my CDS annotations are yellow by default. If I don't like that color, I can make them all purple. Or if I want to highlight just a particular CDS annotation, I can do that here. So I can say, I'm fine with all my CDS annotations being yellow, but I want just this one to be purple. I can click OK. It changes just that color. And then speaking of colors, one of my other favorite tricks to do within Geneious that again might not be immediately obvious is you can color code these folders on the left-hand side. So if I want to make it a bit easier to spot, I can right click on a folder name and do set folder color and change the color of that folder. So it makes it a little bit easier sometimes if you're a very visual person to sort of structure and organize your data that way. You could color them by project or by date, by where they are in a pipeline. All right, with that, I think we're right on time. I do still have a little bit of time though. I can say a little bit later if there are any other questions. Do let me know, right? Send them through the chat function now. Although I recall now this isn't my Zoom meeting, so that being said, we may need to close this down now. Like I said, you can reach out to the support team, that contact us button. Rebecca shared the links in the chat. There's also some internal links you can use. Rebecca or anyone else?
I think there was one more question. So I just wrote in the chat the DISC, the new Data Science Institute at Tufts is having a workshop session this spring and summer. You can check out that website for upcoming data science and bioinformatics classes. I think there was one more question that came through the chat, which was paste alias feature, how to best use.
Yes, aliases, okay. Really have been exploring everything. Okay, so pasting aliases. So what we've been doing here is I've got a sequence, I can click and drag it and this is going to move it to another folder. It moves it out of that folder, puts it in this one. Now there's a few things you can do sometimes if I want to access the sequence from multiple files. I don't want to necessarily create multiple copies of it and have those all independently structured all across my database. There's two things you can do here. One solution is aliases, the other solution is to select multiple folders. So an alias is a way of creating like a hyperlink or a symulink or an alias, whatever term you like to use. So I can have my document here. I'm going to right click and copy. Control C, Command C works as well. If I jump to this folder, right click and I can do paste to alias. So this is creating a link to my other document. So if I change it here, it also changes it. I think we're in this folder. It also changes this copy. Same if I make changes here, that's going to affect this copy. So although you can now see the document from multiple folders, if you make changes, it's changing all of them. So it's completely up to you of what kind of analyses you're working with, you're doing, what kind of sequences you're working with as to when you might want to use that or how you want to use that. The other trick to keep in mind that may also be useful though is if you're selecting folders in Geneious, keep in mind you can hold down the Control key or Command key if you're on Mac and select multiple folders at the same time. So all of those documents from those folders are all going to appear in your documents table. So if this is something you're just doing once or twice and you don't necessarily want to create multiple aliases, that might be a great workaround or might work better for you. So it's completely up to you as to what makes most sense for you. Yeah. Hopefully that makes sense. I can see there's another question that's come through. A local, yes, so Geneious is local desktop software for more heavy operations on the Tufts cluster. I might let Rebecca speak to that a little bit. What I can say is it is a desktop software. You can run it on anything, but it is going to need a graphical user interface available. And like I said, you can import BAM files and that sort of thing. So you can potentially run it in a different place on a server, or you can run your analyses using something else on the server and import the results in. But Rebecca might be able to answer that more.
Yeah, we don't have any interface between Geneious and the Tufts cluster right now. So the only way that Geneious is going to work is on a desktop. I know that there is a Geneious server version, but as far as I could tell, that really takes away from the graphical user interface, which is why a lot of people like Geneious. So it's something I haven't explored too much, and as far as I know, that's not the direction we're going to go in. We do have lots of command line programs that run very well on the Tufts cluster. So once the analysis, my recommendation would be sort of try it out, what you can do in Geneious, and once you outgrow that, then we try to figure out how you can run it on the command line, really making use of the full cluster compute power. We also have, we have a Galaxy server running at Tufts, which kind of is a hybrid between something like Geneious and the full cluster experience, because it has a graphical user interface but actually uses the cluster on the back end. And we have the ability to install a lot of open source tools and even in-house developed tools there. So there are lots of options. I would suggest that if you do have something that outgrows the Geneious program, but you have kind of a workflow that you're trying to, that's working for you and you'd like to move, then reach out and we'll figure out how to set it up.
Great. Any final questions? One last opportunity. Like I said, we've covered a lot. Rebecca, I'm sure, will share the recording or put that in a location you can find it. You're also welcome to reach out to the support team or myself. Support team's usually the best bet, I think. Like I said, they're all PhDs, but you can